Dr. Susan Ustin is going to talk about SAV.
I want to thank you for inviting me to make this presentation. Just to make sure, so SAV is just an acronym for submerged aquatic vegetation. So we're talking about macrophytes, and in the delta, the most common one is Egeria densa. So what I want to talk about in my presentation is what the distribution of submerged aquatic vegetation is in the delta, and whether that distribution is co-located with the water flow velocity, and whether it's co-located with different turbidity levels. And we're also curious about whether the invasion of SAV that took place in the kind of 1990s had affected the turbidity levels. In the delta, we have 4 native species that are submerged aquatic macrophytes. They're shown on the bottom, and we have actually the same number of invasive species. But for most places in the delta, the only common one is Egeria densa, Brazilian waterweed, and when we were doing this study, it was probably somewhere between 50 and 70 or 75% of the biomass. So it's definitely the dominant one. It forms these large mats. The plants are rooted at the bottom, and they grow up through the water column and then float along the surface. And it can form quite dense mats. The history of the invasion of these, in the 1970s, Brian Atwater from USGS had found very few of Egeria in the delta. There were some more in a second study by Brown and Mitch. I can't remember how to pronounce his name, but their studies from the 1970s to the 1990s show very low levels, and then at some point around the late 1980s or early 1990s, these invasive submerged species really took off and reached a maximum. Reached a maximum around 1,800 hectares over the main part of the delta, and there's a few studies that have kind of similar values, including the last one, which was Erin Hester is one of my graduate students, and she much of what I'm going to talk about was from her dissertation. Egeria has both the structural and biochemical characteristics that make it a very competitive invasive species. Probably the main one is the fact that it doesn't undergo a complete dormancy. The plants overwinter, and so when you get into the spring weather and it warms up a little bit, it takes off and it outcompetes the other species in the environment. It has a kind of a bimodal distribution of flowering in maximum biomass in June, and then it kind of dies off a bit, and then it peaks again in the late summer. It's able to do this probably because it has a foliage throughout the canopy. So from the root mass to the top of the stem, it has active foliage. It has so it usually has higher leaf area density than other species out there, and it can take advantage of different light levels because it has a facultative C4 photosynthesis. So it has 2 different mechanisms it can photosynthesize under. So our study, for 5 years, we measured the distribution of these submerged macrophytes in the delta with airborne hyperspectral remote sensing data. So this map shows we measured a slightly larger area between 2004 and 2007, And then in 2008, which was the last year of our study, we measured a slightly smaller area because most of the submerged aquatics were in that more core area. This was a study we did for boating, Department of Boating and Waterways. They are mandated to keep the waterways open, so they needed to know where these aquatic species were located. So the data we used is there was an instrument called Hymap. It's a hyperspectral sensor. It has 126 bands across this wavelength interval from 400 to 2500 nanometers. The visible part of the spectrum is 400 to 700, so it includes the reflected part of the solar spectrum out to 2500 nanometers. That's about the full wavelength distribution of light coming in from the sun. And we collected it at 3 meter pixel sizes, and so over about a 2,500 square kilometer area. So it's a lot of data. It usually took, to cover the whole area, it took 64 flight lines that we had to mosaic together. And with the smaller area, the darker gray, was about close to 50 flight lines. The reason for using this is that with that spectral information, you can get more precise information about what the materials are that you're looking at, either in the water or in the emergent vegetation in the wetlands. So this shows the spectrum. So this part right in here is the visible part. It's very low reflectance for plants because that's where light is being absorbed for photosynthesis, so there's very low reflection. And then higher reflection in the near infrared, and then because these are dominated by water, there's very low reflectance in the rest of the spectrum. And for floating things, they look more like land plants, and so they have a typical plant spectrum. For the submerged aquatics, they look like land plants in the visible and the near infrared, but more like water in the rest of the spectrum. And then water, because it's very turbid, clear water is pretty dark across the whole spectrum, so very low reflectance. With the sediments and algae in the water column, it's much brighter. But you see, it doesn't look like a photosynthetic plant because it lacks the shape of the spectrum. So you can tell the difference between the sediment in the water or on the land. You can tell the difference between land plants and submerged plants. So we also had LIDAR data that was collected over the delta, so we could use that data as well if we needed to better characterize what we could see. So there was some steps of image processing and calibration, producing various intermediate remote sensing products that then went into a classifier. We used a random forest in the R to do the classification. That gave us the highest accuracy. Boating and Waterways and Department of Food and Agriculture gave us about 2 weeks worth of boats on the water each summer to go along with the overflight. So these are the number of ground control points we had. So somewhere between 1,000 and 2,000 points each year. So over the 5 years, we collected lots and lots of field data of open water, highly turbid water, water with algae in it, water with submerged plants, water with floating plants. So with that data, then we could do an accuracy assessment and come up with maps of the spatial distribution of these things. So this is just an example of a map from this sort of central part of the delta in 2007. And everything that's red, not the boxes, but the parts that are mapped red are the actual locations where we detected the presence of submerged aquatic vegetation. And then the two small boxes just show a blow up of the areas. So you can see this area had submerged vegetation. This green stuff in here is floating algae, and we didn't identify it as submerged vegetation. This is for this particular part of the delta for different years. You can see the distributions change a bit. So over the 5 years, it kind of peaked in 2005 and 6, and that declined a bit in 2008 for the total cover over the delta. The average was 1,817 hectares. That's pretty much equivalent to what other people have reported, so we're pretty close. We had about an 80% accuracy for this kind of remote sensing data. That's quite good. And the kappa statistic is another kind of statistic that says, are you doing any better than you would by random chance? And anything over 0.6 is considered very good to excellent. So the mapping was reasonably good. So then once we know the location of these submerged weeds, we could then look at how they're associated with flow velocities. And we had some stations at various locations in the delta, and we used a 1 kilometer buffer around them to identify what was the cover of submerged the SAV around each of the stations, and then did a comparison with the flow rate. And basically, as you might imagine, the weeds are where they're in greatest abundance is at high flow rates, basically less than a half of a meter per second flows, and they do spread out up to higher flow levels, but usually they're very sparse and infrequent. So the high flow velocity events, do limit or control the SAV cover. And this was for 29 stations where these velocity stations were measuring. So we also wanted to look at the relationship with turbidity. And we had 10 turbidity stations, and then we did the same thing. We identified a buffer around the station to compare to the SAV cover. So I got them out of order, but if the station, somewhere in here it's marked, the velocity station, and here's our 1 kilometer buffer around it. So then we looked at the SAV cover in this reach of the channel within that 1 kilometer buffer, and then compared that. So this is, for this point right here, that's what you're looking at. So you can see there's a bunch of SAV in this area, and then compared the percent cover. And again, the channels that have lower turbidity tend to have higher cover, and that makes total sense. They're photosynthetic organisms. They need to have light penetrate through the water column, and so if you have very, very high turbidity, you don't have very high presence of these organisms. And this one has very low correlation because it's basically has no change in percent cover. So we also looked at the relationship between whether in the patches of the SAV vegetation where they form a stand of these plants that are growing through the water column, did they have lower turbidity associated with them? So we sampled at 275 sites, both inside and outside the patch of this vegetation, and we used the depth average turbidity to do the comparison. And you can see that inside the SAV patch, there's more sediment inside compared to the surrounding outside areas. So what you're seeing is, I think, the plants are slowing the flow of water through the stand of it, and so you're getting more turbidity and resuspension within the patch. There has been a decrease in turbidity in the last 50 years. This is data from Dave Schulhammer and Tara Morgan King. And we looked at where you have maybe lower, it's mostly in the areas where there's lower discharge. There's more of a decline. What Dave Schulhammer's data has shown is that somewhere after the drought of the 1970s, there was a flushing of sediment through the system, and then there's been a reduced sediment load or reduced turbidity in the years since then, and that more or less corresponds in time to when the sediment load went down or the turbidity went down. Then that was the period when these plants really took off and became dominant through the delta. So when you adjust it for the sediment input, you can look at changes in the turbidity between, well, we had data between 1975 and 2006, and for these turbidity stations. This is, I say, data, the collaboration with Dave Goldhammer's program. And, if you just look at turbidity levels and percent cover, there's a high correlation with higher plant cover at low turbidity. But the part that's associated or can be associated with vegetation is about, at low plant cover, it's maybe about 25% of the decline in turbidity is associated with those, but it's more common where there's higher density of the vegetation. So it contributes about 20 to 70% of the total trend in declining sediment that's after it's been filtered for the source term. Oops. So you have a part where it's sediment supply dominant and low impact of the vegetation, and then where it's vegetation dominated. So there seems to be a kind of an interaction between the turbidity levels, which are shown in tan, versus the increase in SAV cover over this period of time between 1975 and in the middle 2000s. So this is our conceptual model of what's happening, that if under the high turbidity and high resuspension conditions, you have less light penetration through the column, and you have higher current velocity and not so much invasion of these submerged macrophytes. But under lower turbidity and lower current flow rates, then you have more light penetration and much higher growth and less resuspension, more deposition. So that's our kind of summary of what we learned in that 5 years of working on these. We have, most of this is all published. I can send you the papers. I think in all, we must have had about 19 papers published. Anyway, I'm happy to answer any questions that if I can.
We have time.
Yeah.
I was just curious why you didn't seem to do any measurements in Frank's tract, Mildred?
Oh, well, we did measure in Frank's tract when we were doing the measurements of where the SAV was occurring. Our project was really to map, was simply to map the occurrence of the SAV, and we kind of my student Aaron Hester was interested in collaborating with Dave Goldhammer, and so we kind of added that on as a piece, but we didn't have the equipment to do the turbidity measurements. The reason the SAV cover declined in 2007 and 8 was because boating and waterways had a massive eradication in Frank's tract in that period of time, and did bring down the cover quite significantly, particularly in 2008.
Did it return?
Huh?
Did it return?
Well, we didn't get funded after that, so we had no more flights because we had to pay for the flights out of our project, and so that was when the state, had big budget cuts because of the recession, and so then they quit funding it. My understanding is that it has returned significantly. At the time, I don't know what Boating and Waterways is doing now because we haven't been funded with, and so we haven't worked with them for a while. But they were putting all their effort into Frank's Tract and not paying attention to the plants in the other area. So they were kind of left to maybe expand or extend. But it seems as if most of the habitat they can occupy is occupied. They're not. It's not really changing much from your, total cover. So it's not like they're still expanding into new habitat. They're just more dense or less dense from either from management or from the weather conditions.
I guess until we build more of that habitat for them.
Yes, we could always improve that and make it better for them. But that period from about 1990 to the mid-2000s, they pretty much expanded, from nothing to 1,800 hectares. Yes?
As I listened to the presentation by you and Ryan, I see similarities to the National Science Foundation's NEON program. What's that? National Environmental Observatory Network.
Yeah.
And of course, the motivation for that was to collect data and then store that data in a way that it could be accessible by everyone.
Right.
And then I can see from your presentation, listening to Brian's presentation, that there would probably be some advantage of having a data set where you could pull in Brian's data and he could pull in your data. Of
Certainly.
course, the whole community could do that. But it seems like the way science is structured in the region, the people don't do that. Everybody sort of hoards their own data and then publishes on it. So do you think, just your personal opinion, do you think it would be wise for the region to use the NEON model to have an overall data repository that everybody could access?
I definitely think that would be a benefit because these problems are very complex and they're not controlled by one factor, right? So you need more information. I mean, we've made the image data. We've given it to USGS. We've given it to Joel Dudas at the Department of Water Resources. We've given it to some consulting companies that have been working in the Delta. So we have made it available to anybody that asks us for it. It's several terabytes of data, so it's not like anybody wants to pick it up, but it's definitely valuable.
And I'll say that we are working to get our data up online in real time and working on the data visualization and mining aspects of that as well, taking advantage of some of the tools that are coming out of finance now for doing that. So we strongly agree that needs to be done, and the biogeochemical models that sit with the data, I mean, the data don't interpret themselves, and we need to support that with visualization and processing tools that also sit online and make it available to the management and stakeholder community.
Yeah, I think if, like on this remote sensing data, because they're very large data sets with, 50 flight lines per year, and anyway, maybe putting it on the cloud or something like that where somebody could actually analyze it from online. You couldn't just download it, like FTP it or something. We have to give it to people on hard drives just because of the size. But, I think on the cloud you could do it.